That's exactly what I was putting. It's like 25 or 75 together.
Welcome, everyone. It's a pleasure to have you all here. Thank you all for coming. It's a distinct honor for me to be introducing this year's Carnegie Prize winner. My name is Barbara Shin-Cunningham. I'm the director of the Neuroscience Institute here. I've been here for a year, and I love it here, and I'm thrilled to be able to act in this role as introducing our speaker today, Dr. Eve Marder. It's a end of, we're 4 out of 5 days through Neuro Week here, and this is kind of a highlight of the week so far. Tomorrow we have one more event, the Victor Barg Lecture, and Mr. Barg is here. Oh, I'm pointing in the wrong way. Where is Victor? There. I saw you in that direction. So tomorrow we have the Victor Barg Lecture, and Sophie Scott from UCL will be talking about the science of laughter. I hope you also make time for that because it should be a really enjoyable event. I'd like to thank Deans Dorge and Shinus, both of whom are here, for being here and for supporting the Neuroscience Institute. They're my bosses and they're awesome, so thank you both. Yeah. Yay. And I also want to thank the staff of the Neuroscience Institute who have been amazing at having everything run smoothly so far, as my child says back home. So far, it's been great. So we have one more event to go. We're almost there, but the staff has been amazing. Thank you for all your help. I really appreciate it. Now, I know many of you know Dr. Marder, either personally or by reputation. She will be the recipient today of the 7th Carnegie Prize in Mind and Brain Science. I was misremembering. I thought it was even longer that it had been going on than that. But the Carnegie Prize in Mind and Brain Science, it was made possible, the series of prizes was made possible by a generous gift from the Carnegie Corporation of New York. And the goal of the prize is to recognize trailblazers in brain and behavioral sciences, which certainly fits Eve. The prize not only comes with a lecture, it comes with a student fellowship, and this year's student fellowship is going to Unsel Park. Miss Park, do you want to stand up and just, where are you? There she is, thank you. Thank you. Ms. Park is a Ph.D. student in Alison Barth's lab and is studying the inhibitory circuits in the neocortex and how they change with learning. That makes her a perfect recipient for the prize, the fellowship, because as part of the fellowship, she's being given the opportunity to go visit Dr. Marder's and her lab in Boston, which is fabulous. So again, that's an exciting part of the prize, and there were a number of very qualified applicants. So congratulations, Ms. Park, for winning. Our speaker today, Dr. Eve Marder, as I said, is well known to almost anyone in neuroscience. She studies the dynamics of small neuronal networks. Her work has been instrumental in demonstrating that neural circuits are not hardwired, but actually change with experience, can be reconfigured by neuromodulatory neurons and substances to produce different outputs and different computations. And she's a wonderful advocate for people in science of different genders, of different races, of different ethnicities, of differences in every way. And she's actually been a hero of mine personally since before I had the opportunity to meet her. It also turns out she's married to a wonderful scientist as well, Dr. Art Wingfield, who's in my own field, who's a fabulous person. And the two of them honestly have been wonderful colleagues and advocates that I really have appreciated, and one of the reasons I miss Boston is not interacting with Art and with Eve as much. Eve has a number, I don't know how many of these honors I'm going to read, but there's a huge list. Everybody take a deep breath. Here we go. She is the Victor and Gwendolyn Beinfield Professor of Neuroscience at Brandeis University. She's a member of the National Academy of Science. She's the past president of the Society for Neuroscience. She's part of the Volin National Center for Complex Systems, which is a diverse group of researchers at Brandeis University, united by the common interest of studying brain and intelligence. She's a member of the Institute of Medicine, the American Academy of Arts and Sciences. She's a fellow of the Biophysical Society, a fellow of the American Association for the Advancement of Science, and the list goes on and on and on. She's a pioneer, truly, in this field. She was one of the first people to take computational methods to look in detail at how neurons are interacting and to use computational methods for testing models of computation. The work that Dr. Marder does, as you can tell if you know anything about CMU, really embodies the kind of work we do as well, which is to combine experimental data and models to really understand systems that are complicated. So we're really, really pleased to have her. The final piece of the prize, though, is another really special thing. We every year have Dr. Greg Dunn, who's an artist and neuroscientist, who has now become very well known for his neuroscience-inspired artwork. Dr. Greg Dunn every year designs a piece of art for the winner of the Carnegie Prize. And you see it on display over on my left, on your right. This year's prize is about Eve's work, inspired by Eve's work. When I had the pleasure of telling Eve, or inviting Eve to accept the prize, I got to tell her that she got a piece of artwork from Greg Dunn, and it turns out she has an early work from the same artist from her time as the president of the Society for Neuroscience. And just looking at the new piece, she said this actually speaks to the piece that she already owns, and that's really, really fun and very cool. So Dr. Dunn said he wanted to design a piece for Dr. Marder that commemorates her decades of experience in elucidating the structure and mechanisms of the crustacean stomatogastric ganglion. I didn't recognize them as that, did you? Of course she did, because she is an expert. I did not, but that's really wonderful. The art symbolizes Dr. Marder's achievements in shining the light of her inquiry onto this region, elucidating the functional uniqueness of each of the STG neurons and unraveling the complex circuit dynamics at play. So it's pretty awesome. If you look at the piece of art, as you step from one side to another, the light will pick up a stained cell or not, depending on which. So one of the cells is stained and one is not, and you can see that as you move around the piece of art. So it's really brilliant and wonderful, and I'm super pleased to be able to present this piece of art to you, Eve.
Thank you.
And we are not going to make her carry it back to Boston, although we did threaten her with that. We will actually ship it to you, I promise. So without further ado, I'd like to have Eve take the stage to give her talk, Differential Resilience to Perturbation of Circuits with Similar Performance.
Well, it's a great privilege to be here. First of all, this community is in some ways very similar or emblematic of many of the ideals of the Brandeis community, and so it feels like I'm coming home. Second of all, that is awesome. And there's a story there. Greg called me, or he wrote to me, and he said, well, send me some pictures. So I sent him some pictures. He said, oh, some stained neurons. And he said, oh, yeah, I want to make a scroll, and I want it to be, 6 feet long, I think, he said. And I said, duh, my ceilings are only 7 feet high. That just won't work. And so we negotiated the size. I said, if you make it that big, I'll have to hang it at work in the corridors. He said, oh, no. So we had a little bit of a negotiation about it, but it's really beautiful. And so I thank you. And I will remember this always as I look at it every day. I'm not quite sure exactly where it's going yet. No, not so good. So what I'd like to do today is bring you sort of up to date about some of the, do some of you have to, no, you're not standing, are you, back there? No. Are there people standing? No. Okay. Back bench, okay. So to bring you up to date with the problems that we've been preoccupied with lately. Now, many of you probably know, I spent my entire scientific career working on the stomatogastric ganglion of lobsters and crabs. This is a small ganglion that runs the stomach of crustaceans, and the crustacean stomach is totally different from a human or a rat stomach in that it's got 40 sets of striated muscles that are moved by excitatory motor neurons, and therefore it's much more like your respiratory centers or your shoulder or your mouth than it is like a vertebrate gut. But that said, you might ask, how is it possible that I and other people have spent 45 or 50 years trying to unravel the secrets of only 26 to 30 neurons? And what we have done over the years is the problems we've been studying have changed because I've always kept trying to look for problems of general interest that are best studied if you can identify all the cells and if you have a circuit whose behavior is easily measurable and that you can actually try and really understand how it works. And so what I'm going to do today is bring you up to date with a problem that I think is relevant to all nervous systems, human and even mice. I think, humans are much more like crabs, or at least the human population is much more like the crab population, because it's a wild-type population, not an inbred strain that's been bred in a laboratory. That was only partly a joke, right? And that problem is the question I'll now call differential resilience. And I think, just to presage a little bit where we're going, is you all know, and you've known since you were 2 years old, that every human being and every cat or dog is different from every other individual. But for most of our scientific careers, we've resorted to studying means, and we've sort of believed that if you do things on 15 mice and average, and then report the mean and the standard error, that's going to capture the behavior of every individual that went into that mean. And that's, first of all, those of you who study humans know that that really is very, very fallacious in many ways. But what we're trying to do is put on a rigorous scientific grounds some of the principles that come when you realize that you can have animals or people who show very stereotyped behavior under certain controlled conditions, but yet nonetheless do so on the basis of very different underlying parameter sets. So that's where we're going to be going. But before we go anywhere, I'd like to acknowledge the people in my lab who've done the work. Now, I'm old, so there are a lot of people, and it's only one slide's worth, it's duplicated, right? It doesn't look, yeah, it's small font. So my present lab is up there, including a bunch of postdocs and graduate students. Then there are lab alumni, people who are doing a variety of things, or some of them are postdocs elsewhere. A large number of people who've moved into industry and government, and an even larger number of people who moved on to run their own labs at variety of academic institutions. And I would just like to say that I am exceptionally proud of all of these people, no matter what they ended up doing. And the academic lineage that you see here is actually probably my proudest achievement. I'd like to say one more thing about the people I've worked with, and that is everything interesting and important I've ever done is because somebody in my lab had an idea and took us to a place that I hadn't anticipated. And I think the most important principle is, no matter how smart any one individual is, you're nowhere as smart as the 8 or 10 people in your lab with their intellect pulled together. And so a lot of what I'm going to talk to you about today is stuff that somebody did just because they were smart, and sometimes they did it without telling me. Sometimes they did it, I have a certain over my dead body things that eventually work. But there are a lot of things that people in my lab did without telling me that ended up being quite wonderful in the long run. So I once gave a whole talk about things that people did over my dead body, but I'm not yet dead. But anyway. So these are the questions that I'm going to be coming back to. How tightly tuned do the parameters that govern synaptic strength and intrinsic properties need to be for good enough behavior, circuit behavior, or how different are the underlying structures of normal healthy brains? And then how is individual variability related to resilience to challenge? The last question I'm not going to have time to talk about, which is how is stable network function maintained over the animal's life while allowing for adaptive change. But I'm going to spend a couple of minutes introducing you to there's a clock there who's still on daylight savings time. It's confusing me. Yeah, I know, they're all electronic these days, so why don't they just end? Okay, the system we study is the stomatogastric nervous system. Now, most of the data I'm going to show you today comes from Cancer borealis. It's a crab that lives in the North Atlantic in the area around Boston. We buy them from a distributor who buys them from the fishermen. The distributor keeps them in running seawater until they pick them out and put them in a taxi and they come out the 10 miles or the 12 miles to Brandeis. Anyway, it will become later important that we actually don't know exactly where they were caught, because the fishermen will never tell anybody where they catch their catch, right? The first thing about fishermen is they will lie about where they caught anything. So, and this has become sort of a preoccupation. At some point, we're going to have to solve this problem, because I really do want to know where these animals come from now. Okay, so the crabs and lobsters, because I was working on lobsters, take a taxi. They arrive at the lab. We put them in our holding tanks, which we hold 11 degrees, and we hold them for a little while. And then some poor soul goes down, brings one up to the lab, and we take the stomach out of the animal and then dissect the stomatogastric nervous system off the surface of the stomach. And then we pin it out in a Silgard-lined petri dish, and that's what you see here. The full stomatogastric nervous system consists of the stomatogastric ganglion with its 26 cells in crab and 30 cells in lobster, and then some descending modulatory inputs, which come from these 3 anterior ganglia. They're chock full of neuropeptides, and there are 25 sets of modulatory axons that dump peptide into the neuropil of the stomatogastric ganglion. Here you're seeing 3 sharp electrodes doing intracellular recordings from the somata of these neurons. The somata are about 100 microns, and what you see over there are 2 cells that have been filled. So you can see the neurons with their large somata and the extensive neuropil processes. Those are cells that came from the lobster Homaris americanus, and I know that because they're long and narrow. The crab ganglion is much wider and more diffuse. So we record intracellularly, and at the same time we record extracellularly with pin electrodes on the nerves, and these nerves used to go out to the muscles. So we identify the neurons on the basis of where their axons would have gone, and we record fictive motor patterns that resemble very, very closely what the motor patterns would be if we were recording chronically from the intact animal. So what we see, in so doing, we can record from all 30 neurons or all 26 neurons, plus some of these descending pathways. So we have many, 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 many miles of recordings, of simultaneous recordings from every cell in the circuit. And I want you to know that just having many miles of recordings does not necessarily mean you understand everything. But that's sort of my, one of my general statements. So what is the pyloric rhythm? So there are 2 major rhythms generated by the stomatogastric ganglion. One is the fast pyloric rhythm. The second is the slower gastric mill rhythm. The pyloric rhythm, which you see here, has a characteristic period of about 1 second and is always on in the animal. So like your respiratory rhythm, it's always on, and it changes in frequency, day, night, fed, unfed, all sorts of changes in the rhythm, but it's always on. And that makes it particularly useful for some kinds of experiments. So here you see it with 3 simultaneous intracellular recordings from the somata, LP, PY, PD. LP is a constrictor motor neuron, PY is a second constrictor motor neuron, and PD is a dilator motor neuron. So we have constrictor 1, constrictor 2, dilator, constrictor 1, constrictor 2, dilator, and that is ongoing. So LP, PY, PD. And then the extracellular recording from the nerve, this happens to be a mixed nerve showing all 3 of those units. The nerve bifurcates below that point. You can see the largest spike is the LP, and then PY is the green spike, and the PD are the blue spikes. And it's precisely because we can identify these neurons unambiguously in terms of their anatomical projections that allowed, and that was one of the interesting features of this system when Don Maynard and Al Sullivan started working on it at the end of the 60s. That's last century. Before many of you were born, right? It's a bit daunting to think that people have been studying 30 neurons for longer than you guys have been alive, but we don't have to go there, right? But the goal of the early work was to try and understand what the connectivity or wiring diagram was and how that wiring diagram and the intrinsic properties of the cells gave rise to the dynamics of the circuit. So the people who were attracted to small circuits in the dawn of neuroscience time were looking for preparations that were amenable to intracellular recordings that produced fictive motor patterns that were very similar from the real motor patterns, or similar to, and where you could do simultaneous recordings and then work out connectivity. So this is the connectivity diagram that we had by about 1980. And in this diagram, you see a neuron called the anterior burster. It's an interneuron. It's electric coupled to the PD neurons. There are 2 PD neurons, 1 AB neuron, 1 LP, and 5 to 8 PY neurons, depending on the species. So the AB cell is a burster. It's a pacemaker. And therefore, the PD neuron depolarizes, fires a burst of action potentials, and that continues. The AB and the PD neuron together inhibit the LP and the PY neurons. These are all chemical inhibitory synapses. So this network works because of rebound from inhibition. Rebound from inhibition is the timing cue. And notice there are no excitatory synapses in this network. The modulators present an excitatory upregulation or an upregulation of excitability, but the connectivity itself is all inhibitory. And I think it's very important that in this and many other small systems, as well as in your brain, the rebound from inhibition is a very important timing cue. So here we had the wiring diagram, 1980. Now, if I had shown you today, you wouldn't call this a wiring diagram. Today, you would call it a connectome. And you would say, oh, now we understand how it works. And we knew in 1980 that the original goal was the vain hope that if you had this, you would then understand how it worked. And I can tell you, we had a word model of how it worked then. The pacemaker kernel, these 3 neurons, would inhibit the follower cells. The LP would rebound from inhibition first. It would then hold down the PY. Eventually, the PY would recover from inhibition, turn off the LP, and the whole thing would start again. So that was the 1980 word model. It's approximately still true. But what's missing in that word model and in this connectivity diagram and in every connectivity diagram or connectome that we have is your ability to go from this to dynamics. And if I just showed you this and you didn't know what this was, there's absolutely no way you'd be able to predict these dynamics from the connectome. So this is an example that the connectome is absolutely necessary and completely insufficient to understand how a circuit works. And what's missing is all the voltage and time-dependent currents in each of the cells and all of the strengths and time courses of the synaptic potentials, and then the way in which activity alters the expression of all of those things. So starting in the 80s, we set out to try and describe those properties, to use voltage clamp and other methods to characterize the voltage and time-dependent currents in each of the cells, to try and then be able to really account for the dynamics and their modulation as we started studying modulation. And the first thing that you realize as you start out on that enterprise, that if you really want to account for systems behavior in terms of component interactions, you can do that reductionist path, but at some point you need some way of putting all that information back together again. And the only way you can put that information back together again is to build some kind of computational model or use some sort of tools that allows you to ask questions about how those components give rise to system behavior. And so that's why by the end of the 80s, at the beginning of the 1990s, we started working with detailed conductive space models to try and get insights into how systems dynamics depended on components' behavior. So that tells you how a perfectly well-trained experimental biologist ends up with a lab that's half computational and half experimental. Now I'm going to skip years and years and years of work and show you one of those serendipitous, wonderful things that a postdoc did without asking. And this was Astrid Prince, was a postdoc in my lab in the early years of this century. And Astrid was a theorist, and she came into me one day and she said, I want to build a model of the pyloric rhythm. I said, great. I always said great, and because she was there were a lot of people before her who also said they were going to do it, and they never did it, right? So, I mean, I said, fine, you know, okay. So she set out to start working with some models that we had previously developed for single neurons, and I'd walk by her desk, and she'd be sitting there cursing, and it was all in German, so I didn't understand a word of what it was. And she thought she could curse because she was cursing in German, but there were people around who understood it. So anyway, then she went quiet, and then I walked back one day, and I said, Esther, what are you doing? And she said, oh. She said, well, I decided that it's too hard to hand-tune models, and it's completely true. Nobody should ever hand-tune models ever again. So she said, I thought I would just let the computer do the work rather than me. And so she decided, she took one of these original models, and it had 8 currents in it, 3 potassium currents, a sodium current, 2 calcium currents, an H current, that's a hyperpolarization activated inward current, and a leak. And she said, I chose six values for each of the 8 currents, and I'm letting the computer run all against all. So she was going to run 1.7 million models. And then she said, I'm going to let the computer run them all, and then I'm going to select the ones that look like they're good models for the PD cell, or good models for the LP cell, et cetera. And so she did that from that 1.7 million. She selected 5 models that she thought were good models for the ABPD cell, 5 that she thought were good models for the LP, and 6 for the PY. And then she chose, in the second step of this, she chose either 5 or 6 values for each of the synaptic connections in the network, and then she ran all against all again. And she ran 20 million model networks, which at the time were running on the cluster downstairs. It took a while. But then she was looking for models that would be good models for the pyloric rhythm. And what she did was she started out with the 20 million, and 19% of them were pyloric-like. That meant they were triphasic in the right order. And of them, 2.4% were what she called good pyloric models, because they were triphasic in the right order and fell within a range that Dirk Bucher had measured from 99, these were from lobster, 99 animals that he measured the frequencies, the phase relationships, the number of spikes per burst, et cetera, from all these 99. And he plotted the distributions of all these attributes, and Astrid was looking for all the models that would fall within those distributions, the right set of phase relationships, frequencies, number of spikes per burst, et cetera. So this was the paradigm shift. Instead of trying to find one model that would capture the mean behavior, she was looking for a whole class of models that would capture the distribution of the healthy animals that Dirk had measured. So it turns out the 20 million ended up with 400,000, which were all good enough models for the pyloric rhythm. But then you can start looking at those models and say, what do you learn about what's going on in those models? So when we were writing this paper, I said to her, bring me 2 models with very similar behavior, but that seem to have very different underlying conductances. She came back with the next slide in 2 hours. So since she came back with it in 2 hours, I figured it was easy. You know, you can tell how hard something is by how long it takes your people to do, right? I mean, it's not always accurate, but it's not bad. So this was only 2 hours, which meant that it was easy for her to find, in this case, 2 networks with very similar behavior, but very different underlying conductances. So what you see here is model network 1, model network 2. They have very similar behavior. And yet, if you look at their underlying conductances, you can see that the H current in the PY in this one is big. Here it's small. The PYLP synapse was big here. Here it was small. The ABLP synapse is big here. Here it's small. And so this set of parameters and this set of parameters were totally different. And yet, the network is working very similarly. So this is what I would call a degenerate solution. There are multiple sets of parameters that give rise to a very similar or good enough solution. So I was really excited by this. I was really, really excited by this because it immediately said that some of the variability we were seeing in our experimental and our voltage clamp data was likely real or could easily be real. It wasn't all our fault, right? Because experimentalists, the reason why they plot means and standard errors because they're worried about it all being their fault, right? But I'm making a joke about it, but it's true, right? So, but here you can see there are very, very different sets of solutions. So when I first started talking about this, there was the few random physicists who said, oh, this can't be true because, biology must be optimal. And I'd say, well, since when are you optimal, right? That would usually be the end of that conversation. But the point they made, and it was a really good point, was even though this set of solutions and this set of solutions can both give these behaviors in the unperturbed state, those two sets of parameters are bound to respond differently to perturbation. So then that became the following question, and it's this question that basically has governed the last 15 years of the work in my lab, which is, can 2 or 20 or 100 crabs with different solutions, can they nonetheless respond reliably to the perturbations that they would expect to see? For example, to neuromodulation or changes in temperature in the ocean or whatever. And what happens when you go to more extreme perturbations? So what we've been doing is we've been studying the effects of a number of global perturbations to try and get at this issue of animal-animal variability and resilience to perturbation. So the questions that immediately came up from this first work were, how variable are the parameters in real biological networks? Do perturbations reveal cryptic animal-to-animal differences? And how reliably can animals with intrinsic variability respond to perturbations? So I'm going to tell you the answer, and I'll skip a little. I don't have much of the data, but I think I'll skip it nonetheless. Well, the answer to the first question is everything we measured in voltage clamp or with mRNA expression reveals about a 2 to 6-fold animal-to-animal variability. So we're looking at the same cell. There's one LP cell in each animal. If we look at the potassium channel density in that, it varies in this 2 to 6-fold range. Likewise, the mRNA expression of ion channel genes and receptors varies in that 2 to 6, 2 to 8-fold in the same cell across animals. But at the same time, if I show you recordings from 5 animals, you can see the recordings from animals which would show that 2 to 5 or 2 to 6 or whatever variation in any of the individual components. The motor patterns that you record are very similar. So the motor patterns are more similar than any of the components, which says there have got to be multiple sets of solutions, which invariably must have sort of sets of correlations that can produce good enough behavior. And I'll skip that. So this just summarizes and says that individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when they have very similar behavior. So perturbations. The first perturbation we started looking at was temperature, because it's a global perturbation that is going to affect every single biological process, but do so differentially. And if you think about it, the challenge of trying to understand what happens to a system where the dynamics depend on the sequence, on the voltage and time-dependent activation of a bunch of currents, you might easily imagine that things will get out of whack pretty easily. But temperature is something these animals have to live with. pH, as temperature changes, pH changes, and as people dump nasty things into the harbor, pH changes. We had dead fish in the harbor this past summer, which made me very unhappy. I'm very, since we've been doing this work on temperature, and I live right on the harbor now, I've become preoccupied with the health of the harbor. It's a whole other thing. And then we've been doing some work with high potassium and a few other global perturbations. But to go back to temperature, so one we've done most of. A Q10, we define a Q10 as the temperature-dependent sort of process. I was taught that when I was in college. I was also taught that every biological system had a Q10 of 2. That was a lie. I was taught a lot of things in college that were lies. But mostly in biology, I think. I think the physics I was taught is still probably true. And the math I was taught badly is probably still true. Okay, a Q10 of 1 is a temperature invariant system. Notice there's an exponential in there. So a Q10 of 2 or a Q10 of 3, you can see the process goes higher and higher as a function of temperature. Now, most ion channels, their activation, inactivation, and conductance have Q10s between about 2 and 4, but some have Q10s that are much, much higher than that. In particular, ion channels that are important for temperature sensing or other things have much higher Q10s. So to just illustrate to you what the problem of these Q10s means for a neuron, I'm going to show you a model that Tim O'Leary built just to illustrate the problem. This is a model of a bursting neuron. It has those same conductances we talked about. And at reference temperature 10, all 3 versions of the model look the same. And on the top trace, he has one set of Q10s, and then a second set of Q10s, a third set of Q10s. So these are 3 different sets of Q10s. And in this model, when you go up in temperature, you go from the bursting model to a single spike burster, and eventually it crashes up. This last model goes into a single spike burster mode when you get cold, and when you go hot, it goes into some doublets and then spiky, and now crashes in a hyperpolarized level. And this one is a little bit better behaved. So that shows you that you'd expect that, or it's very easy to see how having, and all these Q10s were in that 2 to 4 range, that having a whole bunch of voltage and time-dependent processes with different Q10s can easily lead to problems. Now, we spent a long time looking for models that would be temperature compensated, and it turns out that they're very difficult to find. So I had a student do a whole thesis on looking for temperature compensated single neuron bursting models, and he eventually found some, but they're a very small fraction of what you find. If you just randomly search in parameter space, they're very hard to find, which is what he did. They are there. Then Tim O'Leary found them systematically, and I'm going to show you at the end of my talk, Leandro Alonso had a way of systematically finding them, but all the solutions for temperature compensated models mean getting a strategy to find a low probability event. And that immediately tells you that my crabs and lobsters that actually do this, and you'll see that in a moment, have found a path to finding a set of solutions that are rare and that are difficult, and therefore there have to be rules that they're using to find them. So what do the biological data look like? This is work that Lamont Teng did a number of years ago. On the top, you see in extracellular recordings, pyloric rhythm at cold, 7 degrees. You see PD, LP, PY, and the same preparation at 19 degrees. And you can see the rhythm is in good shape in both cases, although it's much faster. And the thing that, when Lamont came in and showed me this piece of data, my mouth dropped open, and I said, unbelievable. So what this is the plot of the phase relationship of the turning on and turning off of the elements in the network. And notice that it's flat as a function of temperature. So this is the PD off phase down here, LP on, LP off, PY on, PY off. And notice that although the frequency is changing as a function of temperature, the normalized time of activation, or the phase, is constant. And that's what a motor system has to do. That's what I would have to do if I want to walk slowly or walk quickly. I have to keep the relative timing of that motor pattern the same. And so that's what the crab was doing. It was maintaining phase even though the frequency is changing. So I thought that was incredibly beautiful, and we spent a long time trying to understand how this can occur. And I think we understand it pretty well, but I don't probably have time to explain it to you. But you should think about it. It's a problem. Those of you who are computationally adept should think about it even harder. Okay, that's just another way of saying the same thing. But now, so Lamont worked in this nice temperature range from about 7 degrees to about 23 degrees. And all the crabs, now, the crabs in the ocean, they live between about 2 degrees and normally about 25 degrees. So that's the temperature range in which those crabs live. That's a pretty large temperature range, but if you look day and night, and as they go up and down in the ocean, they see temperature changes. And they can see a 10-degree temperature change in the space of a day. So we have the seasonal changes as well as the rapid changes. However, if you now go to more extreme temperatures, in this case, in the old days, it used to be above 23 degrees. This is a whole problem that where things crash, misbehave changes year to year according to the weather. So I'm living climate change in the lab. It's not so nice. Anyway, this is one animal. This is a second animal. This is a third animal, a fourth animal. And you can see all of them are showing disordered rhythms, and they're all showing different disordered rhythms. And if you think about it, if you say as you go to more and more higher and higher temperatures, and if each animal has a different set of conductances, you would expect them to crash differently and show different phenotypes. And that's exactly what we think is happening here. And then just one more crash. Sometimes they do what we would call a complete crash. So here's 23 degrees over here. And then Lamont went up to 31 degrees. You notice a complete loss of activity, and then back down to 23. So Lamont used to like to say this was a crash, not a cook, because it came back. Okay, so by the end of this, we realized that temperature crashes are widely variable within the population. They're very different from animal to animal, even though the uncrashed preparations show very similar behavior, likely because the animal-animal variability in underlying circuit structures. So what this is saying is that in that permissive range, in the range of temperatures that these animals evolved and developed in, they all found a solution that works. And now if we push them to more extreme ranges, now we start seeing them break down. So the next thing we wanted to do, and I'll go through this pretty quickly, is Sarah Haddad, who was a graduate student in the lab, and she wanted to ask whether altering the modulatory environment could change the temperature sensitivity. And so her idea was that maybe with all the descending modulators present, which is the way Lamont did it, you would see one thing, but maybe if you remove them and then added exogenous modulators, you would actually be able to see different. Okay, I think I'll skip that. And I'll just say, so what Sarah did is she first removed all of, actually, I'll go back to here. So what we can do is we can cut this nerve here, which means decentralizing. And when you do that, the preparations, this is with that nerve intact, and then with that nerve cut, you can see the rhythm has gotten much slower, and sometimes they stop, and sometimes they keep going, but they actually lose the phase relationships they had over here. And you can see this histogram is shifted all the way over in the absence of those descending modulatory inputs. So what Sarah did is she took preparations and she removed those descending modulators by cutting that nerve. And now you can see here 3 preparations, 1, 2, 3. This one had a very disordered rhythm. This one was more or less okay and was now disordered when she went to high temperatures. And this one looked like it was behaving pretty well even at high temperatures. So she decentralized them. Their starting conditions at 11 degrees were very different, and then they responded to temperature very differently. And just a way of showing that, Sarah wanted to use a color code to score the kind of rhythms that you see under these conditions. And so she's going to color in purple recordings that show a classic triphasic rhythm. It's high frequency, low frequency, and all these other sort of forms of, here's an intermittent rhythm, here it's silent, etc. So if I look at those 3 decentralized preparations, you can say the first one was slow and then got faster, but was triphasic and good pyloric rhythm the whole time. The second one started out pretty good and then became disordered only at high temperatures. And the third one started out with an imperfect triphasic rhythm and then changed and made a lot of transitions into different states, including a state of perfectly good triphasic activity that you only see at high temperatures. And so you can see the number of transitions are different for the three animals. And Sarah spent a lot of time actually measuring the kinds of transitions and characterizing them. And then she wanted to take preparations like this and apply modulators, and she studied three modulators. One is a peptide called proctolin, which is an arthropod peptide, which tends to enhance oscillations. The second one was oxytremerine, which is a muscarinic-cholinergic agonist, which also enhances oscillations. And the third was serotonin, which acts on many different voltage and time-dependent currents and basically has a very complicated set of actions. So here's what you see with oxytremerine. Two animals, animal 4 and animal 5. They started out in oxytremerine, really robust pyloric rhythms, and that's maintained as you go down to 31 degrees or up to 31 degrees. You can see everything gets faster and faster and faster, but the triphasic rhythm is maintained. Proctolin, the two animals you see here had very different starting frequencies in proctolin, and that's characteristic of proctolin, but they both handle that temperature range perfectly well, and so that's shown here. The two oxytremerines were excellent pyloric rhythms throughout the whole range, and the two proctolin ones as well. So now let's look at serotonin. Now in serotonin, she had 5 animals here, animal 8, 9, 10, 11, 12. In serotonin, at 11 degrees, you can see how different they are from each other, and a number of them don't start out with a good pyloric rhythm. And now if you go down in temperature, you can see they're all getting messed up, but they're all getting messed up differently. And you can see that in the color-coded preparations. Each of these color blocks looks different from each other, and none of them are showing good triphasic rhythms. So the conclusions from these crashes are that at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation. Each individual animal crashes with individual dynamics, as predicted by their underlying parameter differences. Some modulators may expand the stable range of temperatures, while others make the preparations more temperature sensitive. And I think this is important because we had previously thought of modulators as basically producing variability and flexibility in motor patterns, and now we're thinking that some of the modulators and some of the richness of the neuromodulation that we see may be to actually enhance stability and buffer some of that intrinsic differences and sort of to even that out. So now, I'd just like to show you a few modeling results to end with about this issue of temperature compensation, because we wanted to see what we could learn from building models. Now, I already told you that it's hard to find the temperature compensated models. And it's hard to find them, so we think what the crab is doing is using genetically programmed sets of correlations and ion channel expression and activity to find their way through sort of onto a manifold in parameter space that is a successful solution. So then to find things computationally, what Leandro Alonso has done is he's used some fairly sophisticated genetic algorithms to find a whole set of successful models. So this is the latest version. I'm going to build you a really good pyloric rhythm. So Leandro's models are probably the best. So these are his algorithms. This is just an example of a pyloric rhythm. And in the next slide, he made this picture to say that if he made 15,000 successful pyloric rhythms with frequencies from 1 to 4 hertz, they were phase constant in the same way our biological data are. Now, I'm going to just use one way of displaying the data just to show you the next couple of issues, which Leandro and I came up with. Because one of the things you want to know is if you have a model which has 8 different currents that vary with voltage and time, then you want to be able to see what's happening in these models of different, that are doing basically the same thing, but with different underlying conductances. So here's a model. Here's voltage as a function of time, a bursting model. And now what Leandro's done is on a log scale, he's putting the total outward current up. So this is the total outward current over time. And this is the total inward current down here, again on the log scale. And then in these colors, he's showing you what's happening with each of the currents. So the purple is the A current, that's the fast transient outward potassium current. The orange is the calcium activated potassium current. The yellow is the delayed rectifier. Down here, the green and blue are 2 of the different calcium currents, etc. So now you can see what's happening to all of the currents in the cell as the voltage is progressing. And so now on the next slide, what Leandro's done is he's taken 6 different models, A, B, C, D, E, F. These are models with different starting values of their conductances. And those are all up there. That's that 2 to 6-fold range. However, their behavior is very, very similar. But if you look at these color blocks, and you can just use your Gestalt viewer, you can just look at the purple. This one has a very small A current. This one has a very dominant A current, but the cells are behaving very similarly. Here, the calcium activated K, the orange, again, is changing a lot, and all of these color blocks are different. So you can actually see that even though the final output looks very similar, the mechanism by which that evolves is very different. Okay, so then Leandro wanted to find some temperature compensated networks. So what he asked his algorithm to do was to find networks that were temperature compensated between 10, 15, 20, 25 degrees, but would crash at 30. So here's one example that does that. And I would just like to show you that this particular network is also temperature compensated in phase, because that was one of the things he was looking for. Now I'm going to show you my favorite slide of 2019. Okay, everybody has to have a favorite. This is my favorite slide of 2019. So I'm going to use that color display that we've seen before. And what Leandro did here is he plotted only the last spike of the burst. And now we're going to look at it here. At 10 degrees, 11, 12, 13, 14, up to 25 degrees. So we're going to look at what's happening to the currents over that temperature range. So here, at 10 degrees, the A current is really big. But by the time you go to 25 degrees, it's gotten smaller and smaller. So it's playing a much smaller contribution at 25 than it was at 10. And the yellow is the delayed rectifier, and it's changing as well. But the calcium activated K, which is this orange, is now increasing. So the mechanism of repolarization of the spike, the last spike of the burst, is smoothly changing as a function of temperature. So you're not going through a disrupted form, but the importance or the salience of that current in the burst has changed gradually. And my current way of thinking is that the reason why we have so many voltage time dependent currents in cells is so they can smoothly go through different mechanisms in response to changes in activity. Just going to show you one more thing right here. And that is 2 different models. Here's model 1, model 2. At 10 degrees and 25 degrees, we're just looking at some currents. Here we're looking at the same current. At 10, it gets bigger at 25. In this one, the same current gets smaller at 25. Over here, you've got one of the calcium currents gets bigger as you go from 10 to 25, and here it gets smaller. So these are 2 neurons, which under starting conditions look very similar, but their response to temperature means they're moving through that parameter space in very different ways. Now, I'm just going to end with a very simple comparison I'm going to want you to do again with your Gestalt viewer. These are 3 networks, the red, green, and blue networks, red, that all, and here we're seeing those maps, and they're hard to look at. So let's look at now that red network at 10, 25, 28, and 35 degrees. So here it's completely crashed, and I just want you to look at the color blocks to see what the crash looks like. The next one is the green network. Remember, they all start out perfectly good, but at 35 degrees, this one's only partially crashed. And then the last one, the blue one, again, is completely crashed at 35 degrees, but if I go back and let you look at this last set of colors, that's going to tell you what currents are on and what currents are off. You can see that, oops, in the red one, if you just look at the last, and the green one, and the blue one, the colors are completely different, and that's because they have very different currents in their underlying mechanism. And the very last slide I'm going to show you is something that I think is important for therapeutics. So here we have a model neuron with an A current at 10 degrees. I'm showing, here's the A current is at 100% at 10 degrees, and then Leandro deleted the A current at 10 degrees, and you can see it completely changes the behavior. But then here we have the same model at 25 degrees. It's going faster as you'd expect. But now when he deletes the A current, the model is doing pretty well. So here, the model does well at 100% A current, but the effects of temperature at 10 degrees, deleting the A current, gives you a really big phenotype. At 25 degrees, very little phenotype. Here now we have the calcium-dependent potassium current at 10 degrees, and at 25 degrees, that's been turned off, and it has very little effect. But now at 25 degrees, it's completely, you get a complete loss of activity. So the effect of the same pharmacological perturbation, if you will, is temperature dependent. So just to summarize, I would just say that individuals with similar starting circuit performance with relatively similar circuit mechanism in the control state can nonetheless show entirely different crash mechanisms. And I think this is a real cautionary tale for thinking about disease and for pharmacological treatment of disease, because it tells you that even this amount of information may be inadequate for you to be able to predict how an individual will respond to any number of different perturbations and those perturbations they would see in their normal life. And so I'm going to stop there, and thank you. Am I allowed to take questions? Back there?
So it's very interesting to compare this ideology or the assumptions you're making about what should be studied with what's going on with vertebrates and robots in terms of legged locomotion. With both robots and, to some extent, vertebrates, the emphasis is completely on the mechanical oscillator, and there have been demonstrations that you can generate the behavioral pattern without any neural input whatsoever. So even if there was a central pattern generator in vertebrates, and I can tell you in the robots there definitely isn't.
There is a central pattern generator in vertebrates.
Yes, but its role, from a behavioral point of view, I would argue is very questionable.
I think we're going to have to argue afterwards. I think if you go to any animal below a primate, the role in locomotion for the spinal cord central pattern generators are very clear. It only gets hazy when you get into primates, and that's just because people have been influenced too much by reaching.
But even within vertebrates, if
you get different patterns, but we didn't talk about modulation and the flexibility that you can get when you do that. So there are a whole bunch of sensory inputs that will reconfigure the networks. I don't think there's a problem.
But
I think you,
there will be a basic input from the mechanical system that plays a strong role in determining the stability of the total oscillator.
yeah, so, what's that, so?
Well, that raises the question of, are you studying something that's artifactual? Who cares whether the neural central path generator is stable? Ultimately, the organism cares that the behavior is functional.
Yeah, but the behavior requires, if I take the stomatogastric ganglion out of the animal, there are no stomach movements.
Yes, but then you're turning around and asking the question, what stabilizes the circuit? Which might be the wrong question to ask.
No, it's the right question to ask. For me,
Thank you for a wonderful talk. I'm curious if you have any insights into whether the system can learn resilience, particularly thinking of the modulation.
so learning is a funny word. What these systems all show is adaptation and acclimation. So if you keep them in changed temperature environments, they will reconfigure themselves. The conductance will change. And what happens if you keep them hot too long, their crash point goes up. And so we discovered this. There was the winter of 2012. The ocean never got cold. It was 7 degrees, 6 to 7 degrees centigrade, too warm throughout the whole winter. Now, that next summer, all the squid had gone to Maine, all the dogfish had gone to Maine, and all the crabs that we studied didn't crash up to about 30, 35 degrees. So the whole system shifted. So I wouldn't call that learning. I would call that adaptation and acclimation. But there are the mechanisms, the feedback mechanisms to deal with that extreme temperature were there. Yeah.
Do you think that story is acclimation within individuals, or is it selection?
So we've done two things. We've done acclimation in the lab, where we kept animals for 3 or 4 weeks, and there you're seeing acclimation within individuals. And so we saw a shift. The very extreme thing we saw in the naturally caught population, I have no idea whether the animals that couldn't deal with the high temperatures died, or whether some of the animals went to Maine, and we were studying Long Island animals that came north. So I don't know. But certainly the animals, this is sort of an evolutionary question, certainly the animals that couldn't deal with the most extreme temperatures would have been dead. So we were probably seeing the top end of the distribution at that point. And then the next year, the crash point came back down. Not all the way back down. It's not gone all the way back down to where Lamont had it. So that's a whole other thing. I sort of say, so now what we do is we keep very good animal to animal. When we get it, the temperature, how long we have it in the lab, the day and the month that we get the animals, et cetera. And then we're plotting year by year variations in ion channel expression as well as crashes. And there are certain things seem to have yearly cycles. Back there?
When you're doing these models, do they hint at any limit, like top limit, of what you would be able to adapt to? So these models all will crash around 25.
Well, that's because that's the constraint that Leandro put into them.
So if you put a really high constraint, is there like a limit where the model just won't find a solution, or?
I think there will always be a top crash point, because at some point the K currents will start to activate before the sodium currents. So they'll run into the sodium currents, and so you'll get a depolarization block kind of thing. I don't think, if everything has the same Q10, you could in principle go anywhere. But I think the real question is, I think the question for human relevance is, for those of us who are living at 37 degrees, is 37 degrees a place that gives you the best speed that you can get with a little bit of distance before you start to getting those real crash points? I suspect that there's a limit to what you can do to many ion channels because of the proteins that they are. They can't withstand temperatures above a certain point, and that we're living just below that point, because that's what evolution would find, right? It would find the temperature, I think, that would give you the most neuronal and circuit speed without putting you so close to crash. So, but we don't have much room in there. People become dysfunctional at 106, wherever that is in Celsius, right? Yeah.
Really fascinating topic. So, for those of us who are interested in the genetics and epigenetics of behavior, I'm curious if you've looked at just some like hot-gen sequencing of some of these ganglions, these animals that find different solutions, and to what degree does the genetic makeup of the animal determine which kinds of solutions they go for, or is it more stochastic, where an animal with different kinds of underlying genetics could end up in many different spaces just as a result of sort of ontogenetic.
You know, that is something that would be better done in flies or worms, because there you have clonal animals. And I think what you're asking for is if you took 100 worms and you studied them, first of all, they'll show the variance in behavior. And then if you went into them, you could look at the epigenetics of the individuals and try and correlate them. Number one, we don't have a genome yet, but even if we had a genome, our animals are old. We're studying adult animals, and they're long-lived. So I think you'd want to do that experiment in short-lived animals where you could get them genetically identical. It's the only thing I can think of that, because there's enough diversity in their behavior and in their genetic expression, even in the genetically, and certainly in their epigenetics. So I think if I were really, really going to go after that, you'd probably do it in worms or flies. Yeah.
So what you said at the end, that the mechanisms sort of shifting into in response to perturbation, is that part of the answer to the last question of how the function can be stably maintained throughout the lifetime? Was that sort of
It's related to that. I mean, you have to assume the stably maintained throughout the lifetime. You have to maintain there's some target activity level and then some feedback. And so we have built a whole series of homeostatic self-regulating models. We use intracellular calcium as part of a negative feedback, sort of to regulate channel expression. So the most recent stuff of that comes from Tim O'Leary, and they're very beautiful models. And those models will generate correlations. I didn't talk about that, but our mRNA data shows a lot of correlation in ion channel expression. The homeostatic models generate correlations, and it's almost impossible. It's hard to imagine that you'd find these solutions without thinking about some of the correlations being genetically hardwired. Yeah.
First, thank you for all the work that you have been doing for so many years. I think it has been very influential, and your legacy is still going to be really important for future
Well, thank you.
generations of scientists. Then I had a question actually, I think it might be related to the first question, because I was wondering, what would be the behavior of the circuit under the presence or under the influence of inputs? Because we're assuming that it's producing these patterns.
So the way we're studying here, we've removed all the sensory input, but I have the two sets of answers, which is Mike Nussbaum's lab has worked for many, many years looking at the effects of identified sensory neurons that activate specific sets of descending modulatory neurons. And you can see different forms of the gastric mill behavior, I didn't talk about it before, and changes in the patterns, the pyloric patterns. But the most interesting thing is that when we did the same temperature experiments in vivo. So that's in the animal. We took crabs and we took them through temperature changes. And you see very, very similar behavior to what we see in vitro. And however, and this goes back to sensory inputs, in the in vitro case, they just go up and up and up and up, and then eventually they crash. In the animal, they tail over at a lower frequency, and they go, so we think that what's happening is that there must be some sensory feedback that's sort of putting a brake on that. But at the lower end, it looks, they're superposable. Well, thank you, guys.
Thank you.
It's not 6:45. It's not 6:45.